why don't we start? So you know the drill. First of all, if you've got your video on, you get to introduce yourself. That implies you're a participant. If you're lurking as an observer, you've got your video off. So we'll take that rule. Then we'll go for scribes. Might just ask for ahead of time any input from other groups, because obviously we're coming off the back of an awful lot of discussion. So anything we should be aware of. And then we'll move into the program I think we're all pretty familiar with. So I'm Jason Swedlow, University of Dundee. I run the Open Microscopy Environment. I've already said that to you all. We're very interested in building specifications and open source software for, in particular, imaging data, handling that imaging data, publishing that imaging data, and connecting it with other tools. So I'll stop there, and I'll just kind of go from left to right on my screen. So Nick, you're up next.
Okay, I'm Nick Nystrom. I'm Chief Scientist at the Pittsburgh Supercomputing Center at Carnegie Mellon University, PI of the Infrastructure and Engagement component of HubMap. I think that's good enough for me.
Okay. Thank you. Yeah, Kadi.
Kadi Warner at Indiana University. I'm the PI for one of the two mapping components. And I'm very interested to learn more about these open source imaging data formats, because we have a deep need to get to every single cell, and we need them to have a new UID, but it's a lot of them. So how do you do this best?
Great. Alex?
Jason, Alex Ropolewski. I'm the PI on, first of all, I'm the Director of Biomedical Applications at the Pittsburgh Supercomputing Center. I am also the PI of a data archive for the Brain Initiative focused on microscopy called the Brain Image Library, and I'm also involved with the HubMap project as well.
Thanks,
and
Alex.
I will be a scribe. I'll volunteer.
Oh, you already get a big star on your forehead. If you could just do that. Sabrina?
Hey everybody, I'm a new postdoc at the Medical University of Vienna, studying at anatomy and working on liver mapping. So we are mostly focusing on the natural state of the liver in humans, but also upon diseases and how this is changing.
So I'm going to assume that the other participants, so Jim and Jennifer, don't want to introduce themselves, but if they do, now's the time to do so and to speak up.
Yeah, hi, I'm Jenny. I'm from Stanford and part of a new group that's tasked with data collection for the seed networks with CZI.
That's great. Thank you, Jim.
I'm Jim Haygood. I'm part of the HubMap, one of the tissue mapping centers focused on lung and kidney.
Great, thank you. So Alex has already taken one of our scribe roles. If anybody else wants to volunteer. I should ask, obviously, everybody's got the document, right? And it's where we want to be. There's no questions about that. Right? Okay. Hey, guilt. Any further scribes? Not. I'll try to jump in and support Alex. Okay, so mindful of the various other topics. In terms of the prompts you see there, anything we should be aware of? So in the previous discussions, Kati already highlighted discussion that Alex led yesterday. Any other discussions that we should be aware of or think about linking to or have a look at? Possibly not during this time, but later on, anything that comes to mind immediately?
I saw that in the plenary, you are going to go over to the CCF, the Common Coordinate Framework, and I'm loving that, and I look forward to that session. But for our breakout, I would really appreciate your advice, Jason, and everybody's advice on how to best capture the single cell data at single cell level in different assay types. And I know, Jason, you have deep expertise in that, and I would love to have that as part of the discussion if we have time. because you can't analyze it if you don't have a data format or
Yeah. Okay.
agreement for it.
Okay. Very happy to spend some time doing that. Anyone else? I mean, there definitely are several format discussions, at least I've sat in, I think, 2 of them yesterday. So we can definitely summarize those. And then, but that's not to by any means suggest that the discussions are complete. So we can definitely do some time now. So prompts that we laid out in this topic. So again, and maybe we can together decide where we want to emphasize our focus. So, you know, you've seen the top high level question, how are we going to analyze those data sets? Already Kati is, you know, and obviously I have a lot of affection for this point, but, you know, how are you going to analyze them if you got to have them in a format that you can work with? So that's certainly one of the key points there. Assuming that one could do that, there were several other points that we came up with just in terms of finding those data sets. Even once you found them, you know, how do you browse through them and identify actually or validate that that's data set you actually want? I know that in Nick's case, I know there's an awful lot of interest in making those data sets searchable and accessing that. Personally, I'd love to hear Nick's ideas about that. I think one of the, in our discussions in preparation for this session, one of the things we talked about is, are the data sets that we are coming out of Human Cell Atlas, HTAN, the various other projects, will we just download those data sets? Are they all just going to come on to what are our resources and then we will go ahead and do what we want with them? Or there's some kind of tooling, quote, cloud. I won't get too specific there about a resource, capabilities that will make that the community will be run analytics on. Is that even realistic to do so? I know that's deeply interesting to the HCA project, for example. We talked about other, Peter Sorge was mentioning, for example, in the medical imaging world, the Clara imaging platform for distributed analysis. There's several others across many of the other domains, and even one of the things that I think I would like to do personally is to get concrete data sets out of, concrete examples, excuse me, out of this. So I've already talked too much. I'm going to shut up for a few minutes. Maybe we would, finding and browsing data sets. Nick, do you want to take a stab at that one?
Sure, I'll make a stab at it. So the main thing I'd say first is within HubMap. So of course we have a portal we're building. For those who don't know us, half this call is us, I think, almost. But we're planning our initial data release for June. So there will be search capability there, the ability to find things, download them to do analytics on our own infrastructure, which is a hybrid on-prem and cloud. But looking forward to making things more interoperable, we've had discussions with several other of the consortia, and this is especially around the NIH Common Fund data ecosystem. So we've been talking to various groups, Spark, Kids First, and several others about how to do this. One of the things about HubMap data is, generally speaking, it has less controls on it than some of the other clinical data. So one of the things we're thinking about there is being able to export our data for use with others, such as Kids First, which is children's clinical data, for using their infrastructure for the analytics. How we build those workflows, that's something we're just starting to think about. And that will, expect next year that will ramp up, but we're not really doing any of that yet. Kati and Alex, what would you add? Certainly, Kati, with spatial referencing, that's tremendously important here.
So inside of HubMap, we have currently a division of labor. So there's the data portal, which is search and browsing and filter. And then there is a very dedicated CCF registration user interface, which lets people register tissue cuboids inside of reference organs. And then TMCs, which do that, they can also explore their own data inside of the CCF exploration user interface, just to make sure it's correct. Hopefully in year 3, these two things will merge so that you can also have spatially explicit queries run in the data portal that might not be possible by this summer. Ultimately, I think it's very interesting to learn across the different consortia what kind of approaches exist to actually agree on a common coordinate framework. And we had a really interesting discussion yesterday, and we are trying to get a better overview across the 16+ consortia we have here. But also it's interesting that different organs require potentially a very different approach to a common coordinate framework and to what kind of fiducial points you might be using. So brain, you might use the skull. This is not possible for the heart or for the lung. Heart and lung are moving, they're breathing, they're beating. It's very different from a kidney, etc. So I think it's potentially very organ specific. So I'm going to stop here.
Sabrina, do you want to bring anything in to that? That was very interesting already. So I'm trying, following the patterns you've seen before. I'm listening for the how might we's, and so you can see the green box getting filled in there. Anyway, go ahead, Sabrina.
We actually started now to try to make like a 3D framework. So we are actually doing HREM and CT and MR to get also a visualization. And we are working on the liver. So we try to use the vessel system of the liver to like using it as a reference system to localize really where we are and where we take our samples and where we take our cells for transcriptomic analysis. So this is what we are working on right now. I know it's quite struggling because when we are working with body donors, so quite hard, but we think it's a good point actually for the liver, at least for now, to use the vessel system because it's quite constant, at least for the bigger vessel, so for the bigger veins, to use that as a reference to know where we really are and where we get our transcriptomics from, so that we have a reference that we can then put in a 3D model and pinpoint then the cell transcriptomics to that point. But we are not that far now to see that it's consistent. I mean, we see that for the bigger veins, it's quite consistent with the localization and how they actually are located within the liver. But yeah, but that's our point, how we want to just give a positional information about where we are and where we took our information from. So,
this
and maybe
interesting.
go ahead.
Sabrina, it's really interesting, and I would love to know if you had a chance to see Griffin Weber's recent paper on exploring the usage of the vasculature as a common coordinate framework for the whole body. I put it in our meeting notes under the discussion for our third subtopic, so you should see the link there.
Mm-hmm. Thank you, that would be great, yeah.
And all your comments would be great to hear, and if you can share more on your work with us, that would be wonderful. And Sabrina, I don't have your email address. Could you put your email address somewhere, maybe in the chat log? Or?
Maybe in the blue box where we anyway have to.
Oh yeah, of course.
Yeah, for sure, for sure.
I will do that. Yes, for sure.
Maybe a direct question to both Kati and Sabrina. The finding data sets based on these common coordinates, is that Is that clear how that's going to work? As Kati said, different organs, different systems, etc.
I think it's hard to get a really global system on searching for different organs. It's most likely that you have different reference systems for facial information. So I don't know what Kati is thinking, but I think it would be hard to get like an overall unique reference system where what you are using for facial information.
So I could share my slide if I can, and just show you what we are trying to do in HubMap. So if you see my slides here, so we have that registration user interface. We have a tissue cuboid and you register it inside of an organ. Here's a kidney. And then you have the exploration user interface. And these links are live. You can go there and explore these interfaces. And you can basically click on a heart or the kidneys or the spleen. And you can then browse down to the tissue level. And you can turn on and off certain anatomical areas and even cell types very soon. And this way you can explore it. And then thanks to the registration user interface, you can now take tissue cuboids that the tissue mapping centers extract, and you can register them back into one reference organ. So currently we have male, female of the visible human, left and right kidney, and we have the spleen for both sexes. And so here you see these tissue cuboids registered back into the reference organs. And this could be done for bulk, because it could be that the entire cuboid goes into some bulk analysis, or it could be done for assays which are actually going through slicing, like shown here in the lower left. And then each slice, you have spatial transcriptomics, and then you have hopefully a data format. Jason, I'm going to bug you on that one. So I need a data format for capturing data at this tissue section level, so that it can be reconstructed in 3D. And then these tissue cuboids, they are dealt with in the registration user interface. So we have very good control over that right now. But these other two parts, we need better data formats. I mean, obviously the TMCs have very good mechanisms in place to keep track of these tissue stacks. And there might be slides which didn't work out, or the begin and end of a cuboid typically are not used. But then at some point they have these tissue sections, and now they have the outline of nuclei or cell boundaries. How do we best keep track, given that it's a huge number of cells, and also some cells combined to 3D objects in the middle part of this here?
Hi, this is Jim. It's always a very exciting interface to see, Kati. It strikes me that for each slice, you're going to end up with a box of jigsaw puzzle parts, and putting the puzzle back together for each slice is going to be the challenge.
But we could, for instance, agree across consortia how to best capture the data at the cell level, how to put it back together in these set stacks, which again, every single TMC has a very special and very different way of keeping track of them. And then with the registration user interface, you now have a way to just register it in there. And very soon this will be a hopefully de facto standard. And then you need all three, right? You need the registration user interface information, you need the set stack, which is not standardized right now, and you need that cell segmentation or functional tissue unit segmentation data format. And I think, Jason, on that side, on the cell and functional tissue unit segmentation part, I believe we could suggest a format and just tell people here, use that omnitive, not the other two, and give good arguments for it. And then we just use it in HubMap and hopefully other people adopt it.
Good news is, good news, Trey.
So I'm just kind of seeing how this is going to break down. So first of all, we talked a little bit about how might we find these data. We have a couple of nice examples, which is great. And I suspect, unless anybody objects, we keep coming back to the data format and how we're going to put this data back together. So the second, how might we could be on the data format. So I've started typing that. I'm trying to follow and type at the same time, so which is always a bit challenging. Jim, did you have anything else to add specifically with your specific? I mean, the challenges you're raising are certainly important. Anything else you would add there?
I'll go ahead and put myself back on video since I'm talking. I think obviously we're in a state where we're moving from suspended single cells to more, visual analysis at the single cell level imaging and omics. But in that transition, we're going to generate a lot of single cell data that then have to be somehow registered back to the tissue. And that's going to be, there are higher throughput methods for doing that, but I think compared to the data at the single cell omics level, they're really medium to low throughput methods. Even if you look at hundreds of transcripts, you're not getting close to the thousands that you're generating at the single cell level. So I think that's a real challenge until we get to the ability to just do everything spatially.
So I think if there is some, so pick your organ, but if there is some sort of framework and cuboids or whatever, you have some sort of reference, does that begin to at least give you approximation you can work with, or is there something missing there, Jim?
Yeah, I think I can only speak to lung because that's my organ of interest, right? So there are a lot of single cell data sets out there for lung, and there is tremendous amount of discussion about, what are the different cell types that are identified and where are they in the tissues. And some people have done a good job of, using one or two very specific markers to go back and register cell type to a general location. So for example, a fibroblast that tends to be on the outside of a blood vessel versus a fibroblast that tends to be on the outside of an airway. Or a particular type of, subset of airway epithelial cells called an ionocyte, which is extremely rare but functionally important. Those, it seems, are distributed stochastically within the airway. So let's say you have 5 slices of a cuboid of lung that has an airway going through it. You may pick up an ionocyte in 2 of those slices, but not the other 3. So then where are you going to put it? So I think there's this issue of a, what's actually there, which is what you see when you do spatial transcriptomics versus something that's theoretically present distributed within a some kind of idealized model of a structure like an alveolus. So that's I think really the challenge, is sort of living in that space between what we can deduce from validating single cell transcriptomic data back to tissues using immunofluorescence or immunohistochemistry versus what we actually see in a spatial transcriptomics data set. So I think that's really the challenge, is kind of creating that, this is actually what's here versus theoretically, if you have this type of structure, this is what tends to be here and in what proportion, if that makes sense.
I think so, okay. Maybe we should pull in a couple of those. Jenny, did you have anything to add there? So we're kind of working around a couple of topics here. One is the findability, and one is writing it, recording it, and a representation. I think we're, as we mentioned earlier, we're definitely going to come back to the CCF question later on, because I think it was our sense that all of the topics we were talking about end up connecting into that fundamental question. Jenny, anything from your side?
Sure. I had also previously, before this, worked on the HCA DCP, and as part of the data operations, getting the data through and available in the matrix form. And currently what we are working on with CZI is data visualization in the sense of TISNEs or UMAP plots. So from that angle, just from a data coordination standpoint, what are the needs of the users, and what does it mean to have integrated data visualized between data sets so that it can be useful for the user, if that makes any sense. For example, if you want a single cell ATAC-seq versus single cell RNA-seq for liver or something like that, you know, how is it best to visualize, and how do we enable the scientists to analyze that data?
And do you have any ideas? So I'm sitting here at my desk in glorious Dundee. I need to find those data.
Mm-hmm.
And then I want to combine data that you have with lung data from Jim. Any ideas on that?
Yeah, we, the DCP is, you know, trying to focus on providing the metadata in a searchable format so that the scientists can say, oh, these two data sets are comparable. And also to provide raw data or analyzed data so that you can, in a sense, you know, have your own platform, analyze if you want, you know, ScanPy, SORAD, or whatever. But in that sense, you know, go into the topic of normalization and things like that. Yeah, lots of topics to talk about, but how do we, and from my perspective, is how do we enable that? Provide the information so scientists can do that.
Sorry. Sorry, I'm just trying to type here. Anyone else? Sabrina, did we get you already? Any other how might ways?
I mean, I guess my question with this is that, you know, are we aiming at coming up with a global system, or something that's more localized and more more specific? If we're talking about a global system and we're dealing with different ways of registering tissues, is there a way to resolve those? I don't know. I think it's important to at least discuss where we think we're headed and what we think we can, what problems we think we can address.
So,
Okay, please, Kathy.
yeah, so in our world, and also this is this discussion with Neil Scalenborg, who's leading one of the parallel sessions here. In our world, we foresee that there will be an anatomical view of the world. So most likely a human body, maybe in 2D, but with 3D organs, which are also serving as reference organs, just like what I showed before. And then there will be a kind of spatial or a semantic space where you have different cell types, for instance, as UMAPs or TSNA or other plots. And then whenever you query for a cell type, these cell types would highlight in the anatomical view, but would also highlight in your UMAP or TSNA plot. If you select a cell here or an area of cells here, they would also highlight in the body part view, and that's why it's so important that it's multi-level so that you can zoom in and out. And it would also highlight in your ontology and walk you over to other ontology terms if you wish so to do so. So that's why it's so important to me that we all agree on these cell and functional tissue unit segmentation data formats, because that's the puzzle piece I need, which I don't have right now. And then we can really do that spatially explicit querying. And of course, we're going to use one or another format in HubMap, and we are doing this as part of a PhD project that's going on in my lab. But it would be so much better if there would be de facto standards, and if I would know about them, and if we could all discuss those.
Sorry. Okay, so I'm doing the best I can here with Alex. Alex is doing a great job of sort of capturing comments as we're going. If you look at the bottom of the green section, we have sort of 3 types of how might we's. One is build the resources, and which is centered around the common coordinate frameworks in these different tissues. One is the format, which we've talked about several times, and Kathy brought back. Good for you, Kathy. And then there's a question of comparing these data sets that are nominally related, but are from different studies, taken in different ways, etc. What do we want to do? Do we want to pick what? Do we want to focus on one of those, the format? Because we have to, mindful of the time here, I think we're going to want to, take a short break here and then move to that breakout 2, where we're going to pick one or more of these. And if we want, Costa, are you here to remind us of a break?
Yes,
indeed. Purpose in life?
At the moment, that is my very small purpose in life. It's officially, I just put it in the chat, so feel free to check the note when you have a moment. I won't disrupt for too long.
Take a short break. I'm going to stay here and kind of mull this over. So if there's anybody who wants to discuss this, that's fine. But I'll take a short break and start again at 20 after the hour. So that's about, I think that's about 11 minutes or something like that. Is that okay? Give people bio breaks and all that kind of stuff. And I guess come back ready to ask, okay, which of these do we want to take forward in terms of a problem? And we have the remaining time to define one or more of these. Okay?
Thank you, Jason. Sabrina, where did you put your email address? I can't find it anywhere. Maybe we work in different documents here. Or just put it in the group chat. I would love to follow up.
No, I just didn't do it yet. I'm sorry.
And Jason, do you want us to put our names down here in the 17.c?
Yes, I think that's where we're going here, Kati. So, yeah, do that now, later.
Yeah, I actually worked in the 17.j topic master, so that wasn't useful.
Yeah, it gets a little confusing. Sorry about that.
All right. So Jason, do you know other consortia which have agreed on how to best exchange cell and functional tissue segmentation results? So it could be by human person, could be by an algorithm. I don't care about that right now. But every single TMC seems to use a different data format, and I'm not the one who should tell them what to do. I just don't think this is my role, but I need to have one format. And so I'm looking to you and to others to help me with that.
Yeah, so, well, with respect to segmentation, which you brought up a couple times, and which is obviously pretty important, OME has its own representation. One that I'm surprised it actually worked out. I think it's pretty advanced. Is in the EM world, so they have a dire need for. It's as always in these things. It's the same thing. It's just different. So there's a more enormous amount of differences, but ultimately it's the same thing. So they have this idea of subtomogram averaging. So you take a tomogram, an electron microscopy, effectively a 3D data set. Details don't matter, but then you have to calculate subregions of that. And they needed a way to do that for their public resources. And so they've come up with something, a representation of segmentation. ImageJ World has done something similar. They have their own representation. Certainly, so when you say each of the TMCs have their formats, you're really referring to the imaging data that's coming in from the TMCs, if that's what you're referring to?
So ultimately, we are discussing, should it be a TIFF with multiple layers, one layer per cell type, because this would be very efficient, especially if you tile it up and zoom in. But then the TMCs are pushing back. They want really to be able to address every single cell, not just the same cell types, which initially we thought this would work. You know, you can turn on and off in your ontology a certain cell type, and they would be visible on the anatomical view, but also in the TMC UMAP view. But I think they really want to get to every single cell. And then, of course, that causes a huge minting of UUIDs on the on Nick's and Alex's side. So there's a big question there. Can they do maybe 10,000 cells this year and 1 million cells next year? Is this feasible on their end? And how do you deal with this in a web browser? So even TIFF versus SVG kind of formats is not clear. And just because we are in a break, I can maybe show you what we are doing right now, and you can comment on that and say, yeah, this might work, or then somebody else had a better idea before that. So in our world, again, we take, can you?
Got it. Yeah. I see what you're doing.
Here, yes. So here in this one, so we have a high resolution sample image from the protein atlas, actually. We use this as a reference system, as a base map. And then we have one layer, which is the nuclei, and then the other layer, which could be cell vessels. And this way we can also get the distance between cell types. Yeah. But we can also have, instead of cell nuclei, we might have cell boundaries. And so we need to capture this kind of data. And one way of doing it is to just have multiple TIFF layers using the one preferred omniTIFF format. And again, here in this particular example, you could then have segmentation boundaries from machine learning or from manual annotation. You would have then the different layers, and you could turn them on and off based on types and subtypes and states and substates, etc. And this would just be TIFFs, which you can tile up very nicely. But then again, I think many of the TMCs really want to see an SVG format. And so it very much depends on what kind of software they're using. Based on that software, we get different data formats. And the softwares are not even having the same reference system. You know, in some cases it's in the top left, in some cases lower right, and in some cases in the mass point. So it's just all over the place. And I don't want to bring order to this. I just want somebody to tell me, yeah, use that one. Somebody spent their PhD on this. I want to use that, right?
Okay, so, and then the problem is that for one of your layers, for example, nuclei, TMCs will want to subdivide those nuclei into n different cell types, right? And so
yeah. And that's not a problem. So we would just have different TIFF layers, or we would have SVG polylines, which have metadata, and it can be long metadata. It can be 1,000 fields. We don't care.
So maybe we need to wait for Nick to come back, but I think, you know, in the how might we have a let's see, so this is distributed analytics, and so but the key is we've got two data sets from two different parts that we want to bring together. So they will have to have, yeah, I know. Sorry, and different data, sorry. And if you do what you are talking about with a TIFF-based model, Nick is going to get unhappy pretty quickly because you have a significant inflation in the number of masks that you're adding to each image.
And then tiling also, right?
Right. Oh yeah.
So I think he can then showcase what supercomputing can do for us all, because he can pre-compute this all for us. And I potentially believe that Nick would actually be happier with a TIFF as opposed to minting millions of UUIDs for single cells.
I'm sure that's true. One of the things that we're working on, so we should definitely have a discussion. So the people in your, we've talked about this a couple times, the people, we should get our groups together. We're actually in the process. So I'm writing proposals kind of as we speak. And also we're writing project plans to start work on this. The problem with the TIFF-based view is it will work for that two-dimensional case, but the moment we get into something that's 3D or multi-channel, it becomes, effectively you have an artifactual fixation with a specific reference position of a specific 2D plane that has nothing to do with the biological system and have everything to do with the physical position of the detector whenever you took the data. And so it's fine, and we've lived like that for an awful long time, but it's kind of running out of steam. And so there are several efforts to look at different binary representations, which then could have associated with it n different masks or derived representations, which you could, as you're saying, toggle on and off or search through, etc. And each of them need to have metadata connected to them, and quite possibly, as I think you're hinting, some number of unique identifiers to define them and be able to access them. So when I want to access that data structure of the nuclei of cell types Q, R, and Z prime, I don't have to suck down 100 terabytes of data, just it's an exaggeration, but a substantial amount of data to just get to the point I want, right? So that's exactly what we are working on. And that means, it ain't done, but the good news is that there's a substantial amount of work already. So I'm not sure, I mean, there's a bit of I have a bit of a conflict here. I mean, well, you and I think have a bit of a conflict here. We can either make the rest of the session about that, or we can agree to, I think we'll have people joining us back here in a second. But,
let's make another meeting on this, Jason. I would love that. And I would love to bring in Bruce, who has masterminded much of the data structures we are using right now. Again, it's not our place to develop de facto standards in that area. It's not our area of expertise. So I would love to benefit from your work.
Yeah, so it's these are the good news is that these are common problems, and they're creeping up in all sorts of spaces, and imaging is a big one, but it's not the only one. And there are several people that are converging, and several different fields that are converging on, first of all, a binary representation called ZARR, Z-A-R-R, which you may have heard of.
Yeah,
and
it's Niels's team,
Yeah, but there's more work to be done that needs to be done to be able to adopt that. It'll make Nick and colleagues happy because there's libraries that will support representing that data on object stores, so cheaper storage mechanisms, etc. So, hi Alex, we're just talking about ZARR and things like that. So
okay.
Just having a bit of a side conversation.
Yeah, I've had my guys start to look into that, and their first comment was, "It's complicated."
Yeah, I mean, the real answer is it's not done yet, right? So all of the things that everybody's used to having aren't there. So it is 21 past the hour. Well after sunset here in Scotland. So everybody back? Nick, you with us? Sabrina? Kenny?
Yeah, I'm with you.
Okay, okay. So Brendan's here. Give Nick about 30 more seconds.
Alex, I'm inviting Bruce to a meeting with Jason and myself. Do you want to be in the ZAW discussion as well?
Poor Alex.
Maybe not. I hear a no.
Actually, so there's two guys that work with me on the Brain project. One is Greg Hood, and the other is Art Wetzel. Both of them, particularly Art Wetzel, has dealt with a complete variety of different types of imaging file formats. He's come up with different ways to virtualize file systems so that image access is faster and whatnot. And really, those two are the ones that are most appropriate folks from my group to participate in those discussions. But since everybody's working from home, I don't really know exactly what they're doing at the moment. But you can send me the notes, and I'll see if they're available at that time.
Cool.
All right.
I will do this. All right. Thank you.
Cool. All right. So, colleagues and friends, we are now down to breakout part 2. We're going to focus on building resources for finding these data sets. Do we want to vote on this? We have how we might build the agreed data format, compare different data sets that are nominally related. Did I miss anything, first of all? Anything that we want to add there? Take that as a no, but if not, start typing or shout or do any of the different means that you each have. Resources for finding data sets, votes? Or maybe why don't we do it this way? Let's go around the screen and ask which one would you start with? And then if we can get that one done in the remaining time, we can work on a second one. So top left of my screen is Kati. So is it obvious, Kati, or what do you do?
As soon as I have that one more puzzle piece, how to best keep good track of cell and functional tissue unit segmentation data and these set stacks. As soon as I have this, and everything else for me falls in place. These two pieces are really critical. Everything else seems solved for me from my end, but maybe I don't know your use cases, right? But the use cases I have seen inside of a HubMap, where somebody has one cell and wants to understand what's in the neighborhood of that cell, what's the distribution of other cell types. Or I have two cells and I want to know the distance. Or I want to have an ontology term and I want to search for it and see it not only in the anatomical view, but also in the UMAP or TSNA view. All of these use cases I have seen there, they become doable if I have these two data formats. That's why I'm hammering those.
Okay. So I've just highlighted what I just tried to type, Kati. Is that, how might we track? Sorry, just typing too quickly. Is that a good summary of what you just asked? Well, please type away.
I'm gonna
Fix it. Kati's typing. Alex, you're next on my screen. Which one would you pick?
I don't know. It's a tough decision here. So, I mean, there are so many issues related to all of these things. Can I pass? I mean, if I was forced to choose, I would say maybe. Oh, geez, this is tough. I mean, I see the need for all of this. The agreed upon data format. I'm wearing my Bill hat, and I'm seeing, yeah, that's, you know, with different types of data, both, you know, spatial transcriptomic data that we're now getting, and you know, anatomical data from different technologies. I can see the need for that, plus the way people want to analyze the data, compare different data sets that are nominally related as well. I see. I'm going to go with number 2.
Okay.
The data.
Got another vote there. Jim?
Yeah, I'm out of my area of expertise because I'm not really primarily a data person, but from a sort of a potential user perspective, I think deciding similar to what Kati presented earlier, different ways to enter the data and ways they can be linked, right? So you may be interested in comparing 2 cells. You may be interested in comparing 1 cell over time. You may be interested in comparing 1 cluster over a longer time frame, such as aging. So trying to think of once you enter with a particular question, what are the data sets you need to link to make sense of that, and how is that made in a way that's friendly to the user. That makes sense.
Voting for a second bullet point there, although there's an awful lot, obviously there's an awful lot of requirements there. Data supports the data types, the experimental issues, and also it's accessible to the user. There's an awful lot there, which we all know, but okay. That's representing your vote appropriately. We got 3 votes for the second. Nick?
I think, so I'm also inclined to agree with the idea of the second. My only fear is that whenever we talk about developing a de facto standard is that years are lost in deciding on the standard. So I think I would rephrase it as a mechanism for interchanging data representing all those things. I'm mainly thinking here too that we're dealing with a lot of consortia that some are young like HubMap, some have been around for quite a while. And I think if we worked on a framework such that, let's say people had single cell data, or they had whole slide imaging data, or they had something else, it was sufficiently self-describing in JSON LD or whatever, that we could at least do a conversion where we had to do some point-to-point analytics and facilitate that translation. I think trying to get a single standard is going to be a long exercise in frustration.
Are you asking how can we identify the concrete examples that would exercise these concepts, but also give us a sense of making some progress? Am I putting words in your mouth there, or?
Yeah, that's the gist of it. And I'm really thinking, I think things have to be made concrete. If we try to solve the general problem, it's just too big.
Okay.
I think we have to look at concrete examples of what we might want to analyze in a distributed sense.
Okay.
And let's say we want to analyze single cell data from HubMap and some other NIH-HCA. Let's make it easy.
Okay.
I don't know whether or not we have the same formats internally. I doubt it.
Mm-hmm.
But could we provide just filter layers that scale as n rather than n squared with those numbers of formats to allow that interchange.
Okay.
And that I think we agree on eventually.
Okay. Sorry. Okay, next on my screen, Sabrina.
I mean, I agree with Nick, but I think we should maybe also go into the importance of the anatomic structure, and to see really also in context with what you see in the data set and in context with where it's located in the organ, because this information might also help you interpret what you see in the data sets. So this backtracking back to the organ layer, I think it's really important to get it yourself to the point that you can really interpret what you see in the transcriptomics or proteomic data that you provide. So.
Okay. If we take Katya at her word, we're going to get that with kind of the first issue. We're going to register that tissue cuboid in some way. That's what we're referring to there, I think, in that first requirement. Am I correct, Katya? Is that? Yeah. I think you're muted.
I can put the link to the current registration user interface into the document.
Cool.
And again, currently it works for kidney, for soon for spleen, and then we are working on also making it work for heart, lung, and colon. So I think it's really across organs.
Okay, Jenny.
Yeah, I think just what everybody has really said. Just to shed some insight on what the HCA has. We actually don't have that much visual data or IHC or anything like that. At this point, it's just mainly focused on transcriptomics that we run through standardized pipelines to get an expression matrix, which as an option could be downloaded as a ZAR file or a Loom or CSV. And yeah, that's the current state, but you know, I do agree it's difficult to have one standard, but be good to be able to at least understand and convert if possible, if needed, minimally. Currently metadata is JSON, so yeah, that's what we have.
Okay. Well, thank you all. I guess that pretty much decides that. So I guess what we want to do here is to, so we're not time for another break. Don't tell me this, please.
No, it does.
Okay.
Not another break.
Okay.
Has your group been able to get to part 2?
Yeah, yeah. So I think what we probably want to do is the second bullet point, which we've agreed that's what we're going to focus on here. We want to copy. I think there's an awful lot of information going in there. We want to move that down to the excuse me, the box in part 2. Is that correct? Does it make sense to everybody? So we're working, but working still in part 1.
Yeah,
so.
yeah. I think copy that whole second bullet point.
Okay. Thank you, Alex.
I will do that if people are done editing that.
Okay. All right. Let Alex do his job here. Let me type all over him.
So Costa, are you going to bring us high tea at 3? Is this what you're?
Excellent.
I'd be happy to.
I could use a gin and tonic.
I just need I just need the airlines to get all the planes back up in the air, and
we're well into cocktail hour here.
No sweat. Pardon my interruption. Just put some light notes in the chat for you, but it's all in the Google Doc, and you're in good hands. So.
Okay.
I'm available if you need me.
Yeah. We're getting all the way to the next step. So I think, let's get, mindful of some of the things that have been said, I think in various ways by each of us. There's a bit of if I can speak out of turn here, there's a bit of impatience to get something that's concrete and that will be usable and can effectively solve some of these problems without trying to be the all things to all people, which I guess is a bit of what Nick was saying earlier. So, ideas on how to do that? I'm going to just speak out of turn here. Concrete use cases of just one or two things that have a biological significance are, in my experience, a great way to go, a great way to start. But that's an opinion. I happen to be shut down.
Yeah, so Jason, I agree with that. One thing that we had punted on for right now was really looked at such use cases with HCA and HubMap. We have this new Common Fund data ecosystem. There are calls for proposals. The next one is due in May. And we intentionally did not track out what we do with HCA until this meeting. But I think we could figure out something very concrete. Just a specific HubMap HCA thing that you would do that would help to answer some medical question. That could be extremely valuable. I know how to build infrastructure. I know how to build analytics. I don't know what that medical question is. So I need help with that.
Nick, can I ask you to put the relevant calls and a little bit of information into the green box we're working in? So,
sure.
Just so everybody can see that.
I will do that. Give me a second. I have to find it.
Alex, I think we got some background noise coming in from somewhere. So. Okay. Any others? Any other ideas? Actually we've got a significant number of projects represented here. So any other concrete examples? And here, and I'm going to speak again out of turn, something as simple as mapping kidney in a kidney experiment in, for example, Human Cell Atlas to HubMap or whatever, or connecting across two of the projects. And you guys will have better knowledge of the specific experiments that are available to do such a thing. But I don't know if that's a terrible idea.
I think one of the unknowns on my behalf is I know what we're doing HubMap wise, but I don't know what they're doing HCA wise. Are they capturing exactly where the tissue sample came from, and how are they doing that if they are?
For the data sets that are currently in the DCP, I would say that information is captured just using ontologies, describing the biosample that was derived from the organ. So yeah, like I said before, we haven't really integrated any of the spatial data yet, so nor the CCF, but this is just talking about the DCP data sets, but for the HCA general, I'm sure people are currently working on that as well.
How would we find that out, Jimmy? Or anybody? Jim, Sabrina, anybody? Who do we ask?
I think a good example we can look at was the initial plenary session on COVID-19 and looking at the receptors for SARS-CoV in the airway and lung. This was an effort that was done very quickly by an international group with representatives from multiple different consortia. And they did somehow at least give some anatomical framework to that. In other words, some of the data were from upper airway, some were from lower airway, some were from further into the lung. So that's a very kind of gross example. It's not down to the street level that we want to get to, but at least they were capturing across many different consortia, capturing some spatial information.
So the action there would be to find out what, I've heard anecdotally in a couple of these meetings that none of that was very easy.
No, I don't imagine it was, but there may be some important lessons learned that we can get from that group. That group did a lot of things in a very short period of time, and maybe they haven't even had time to look back and see exactly what they did and how they did it, but I think there's a lot to be learned from that.
Okay, so it's an action to contact.
I think something else that's kind of coming out in my mind in thinking about this is there's some organs and tissues like brain that are very structurally defined. In other words, you know, what's going on in the cerebellum is very different from what's going on in the hippocampus, whereas other organs and tissues are a little bit more modular. You know, the kidney has repeating structures that occur over and over again throughout the organ. The lung probably does that as well, whether, you know, other organs, the spatial differences are more unique to each portion of the organ. So it may be worth thinking about that, or is there some complexity that can be reduced by creating a prototypical structure for an organ that's, you know, a unit that's repeated multiple times versus an organ where each anatomical area is completely unique.
Oh, good ideas.
I would say that's also partly possible in the liver, where you have different segments, where it's where you have a defined area at least which you can separate, and it's also postulated, for example, that these segments have different functions and comparison to their distance to the veins, so the main veins like the vena porta and the vena cava, where they get the oxygen supply and the nutrient supply and differ there in the function. It would be possible actually to at least let's say separate them and define them and compare that data.
So some candidates, but sounds like we need to do some work to identify data sets, consortia, labs, et cetera, right? Kati, in the development of the in the examples that you've been building, are there any cross consortia or cross project examples that you have access, that you know of or have access?
So we just got one of those KPMP glue grants, and that will give us access to high quality 3D data by Zed Winfried, who is in KPMP. And so we will bring that into the exploration user interface. And by the way, inside of HubMap, we also have some data sets where we just know it's kidney or it's heart or it's lower part of the spleen. And because we have 3D objects which are annotated with those terms, we can just put it in the heart. So if you click on the heart, you will still see that data set. We don't have it registered using the registration user interface. We might know its book and will just be registered at that level for which the information is available. So this is not a problem at all on our end.
Okay. Oops, sorry. Didn't mean to put that there. Good. So you're suggesting even within, for example, HubMap or presumably HCA or others, there may be some examples where we can pick these up.
It's my understanding that if you go to the HCA portal, the Human Cell Atlas portal, you can search for human kidney and you will see, I think, 2 data sets there. And then you can search for human other organ and you will see a bunch of data sets there. And we could totally take the human torso with some organs inside. And if you click on the heart, you get to see all the HCA and all the other data sets. And if you click on another organ, you get to see their data sets. As you might have seen in the CCF session yesterday, we started on this larger table with all the organs and all the consortia, because I wanted to understand who's working on what, and it's so many working on so many organs. And so again, I don't yet know where they all have their data, but for some of them, I know where their data portals are. And I now know from the experts themselves what kind of organs they are working on. And then yesterday's effort was very much on agreeing on these anatomical structures and cell types tables. And that's a different effort yet altogether again. I'm not going to touch this here because I think we have this under control in the CCF part. But this is an attempt to have people agree on the names for the anatomical terms and cell types, which is another puzzle piece I need in my work.
Great. So mindful of the time. So Nick, I've just taken the CFDE grant info you pasted in above. That was sort of in the first part. I moved it down in the second part just so that it's. Is that okay?
Yeah, absolutely. Too many windows.
Yeah.
Yes.
Yeah. No, I know. Okay.
Well, yeah, it's so, because the kidney appears in each.
Exactly. So we have some work to do to identify a couple concrete examples. And here we're just trying to go from, if I understand what we're suggesting, from kidney to kidney or whatever, from glomeruli, I guess, to glomeruli, across either projects or even, as Katya was just saying, within projects. We have a couple ideas here. Katya mentioned sampling of other projects in the, if that's the CCF breakout, right, Katya? So we can check those notes, right? Nick, are you suggesting we try to write a grant together? Is that the...
What a brilliant idea.
He loves writing grants. Nick loves doing that. That's his...
I would say I love it. It's what I end up spending my waking hours doing.
And he's good at it. Awesome. It's good.
Yeah. He's got that beautiful sunset behind him, so what else is he going to do?
Right? Yeah, wishful thinking. Yeah, so I know there are things we have to do in May, and we've been assured there will be additional calls for these common fund activities as well. So it doesn't necessarily have to be in May, but for some time in the future, I think that'd be a valuable way to get started. I think one of the challenges with this HCA meeting is that because of all the breakout rooms with our virtual format, the chance of having all the right people in any of the small rooms at once is pretty small.
Yeah.
If we establish that as a goal we'd look for and then follow up after this meeting.
My response to what you just said is I think you just volunteered for one of those 2-minute soapboxes.
Fair enough.
And
I won't take 2 minutes, but fair enough.
Yeah, exactly. I mean, that's probably a 1 or 2 minutes. Okay, so I apologize, between all this, I haven't read any of this, but I'm just gonna, sorry, I haven't read any of the Nick's links, and I'm sure I'll look at them later. But some kind of proof of concept testing to develop a data format to actually mediate this connection between two nominally, two independent data sets. Is that, from what you know, Nick, is that in scope, or is that the kind of?
It is, and we've talked about some aspects of that with Kadi and with a Spark collaboration with other folks there. A lot of that was on, well, I think it was more zontological issues and spatial registration. That's one aspect. With some other consortia, it would be more around how we exchange and go back and forth from bulk genomic to single cell fine grained genomic data. There are a lot of questions we can describe here. I think where we could get some medically interesting questions around multiple of it, that's where we were with both of these. And so far, I've put those off just because we had a lot of balls in the air at once. And we've personally been very busy with a lot of the COVID-19 modeling as well. So that has been taking a lot of my energy is supporting that. But certainly for May and beyond, identifying areas where we could do this across even more than 2 consortia, but around maybe a single data type, around a single medical question, can be very valuable.
Okay. Okay. Anybody shaking their head? Anybody thinking, rolling their eyes, or thinking, oh, another grant? What a waste of time. And anybody having any allergic reactions or anything? So I'm starting to type into the final template. I very much appreciate. I guess we have more or less 3 sentences. Nick, if you want to, so the point you just made, I agree. I mean, it's great having these discussions. On the other hand, the fracturing is a little bit difficult. So in that blue box, what additional expertise do you need? If you want us, you know, I think we can start filling that out. You want to take a few minutes to just to fill out this blue box?
Yeah, that would be good. I mean, my thoughts on this too, before we can answer the question about what additional expertise do we need, I think we need to define exactly what we're doing a little bit more. Are we just targeting kidney and lung, for example? In which case, you know, the additional expertise may be people from those groups that are dealing with that. Is it, you know, are we really focused on formats and exchange? Are there experts that we would need to be able to contribute on that? So I think we need to kind of identify a little bit with a little bit more specificity what we're doing before we can tap what expertise we know we're going to need.
So I would flip that a little bit, Alex. I would think more in terms of, so you're aware of the things we've been doing with Katya and Bernard on Spark and spatial registration and nerves and the like. I think more in terms of what medical questions might we answer by putting a couple of these consortia together. And then that would drive what the organs would be, what the tissues would be, and what the data types would be. Because there's a lot of things we could bring data together to do analysis, but I would like to know what question we're trying to answer first.
Well, but the other flip side of that is, you know, if we're going to, you know, get some medical questions that are focused on the liver, for example, then we need to know what liver data sets and who's doing what liver-wise to be able to pull in. So I think it's kind of one of these chicken and egg problems where yes, there's some, you know, medical questions are very broad, and a lot of times they're really not necessarily organ specific, because there are some questions that would, you know, you'd be interested in looking at multiple organs for. I don't know. What do other people think?
I think, you know, the table that Kati helped develop and pointed to earlier, where she's trying to capture all the different consortia and all the different organs and tissues that they're looking at, is probably a good place to start. We could see where the most data and the most types of data are, and then look for ways to link those together, either around a specific question or around a specific task, such as, you know, registration to anatomy or that sort of thing. But I think there's already a lot of data emerging, and probably working with where we have the richest assortment of data sets is probably a good place to start.
So I've tried to type the name of the problem in the blue box, and very conscious of being abridged, not by passport, but by funding mechanism. And so it's okay here to actually to state we are going to develop a piece of technology to solve a problem. I'm aware that a lot of times in the US funding system, that's not always possible. So have I captured, so question one, have I even captured roughly what we're talking about under this problem we think we can solve? And is that even a problem that is fundable under any of these funding systems? And like I said, I just haven't had time to look at Nick's proposal here. I mean, obviously there could be others that we need to look at. We're looking at that one for the moment. There could be others.
And Nick, I don't know if you can answer this and say just no if you want to, but I believe because of COVID, we might not see much new data in the next year of HubMap. So it could be interesting for us in HubMap to see what data we can get from other efforts, which might already be older than HubMap. And so it could make sense to use some percentage of our budgets, our collective budgets, including mine also, to look in how far data from HCA or from other consortia is valuable for the HubMap mission. And in this moment, then we could potentially be interested in having these cross-search and cross-analysis and cross-visualization efforts in place without even looking for additional funding.
That's definitely true. We've actually been encouraged in the past to somehow, in no clear way, incorporate HCA data. And I think you know that. I do think that would still be one of these common fund projects because our funding that we're looking at for year 3 of HubMap would not necessarily be enough to do that plus what we already have to do. So I think it'd still be a good common fund project, and noting that those are small anyway. So I think that would still fit. I think you make a great point though. There's bringing in the HCA data, and there's also the various common fund projects that are sunsetting. And this could be a really good time to think about federating some of those in ways to be mutually beneficial.
Which ones are sunsetting?
So I think GTEx is one of those. I think they are within their last, I think they end in 2022 or 3, sometime, I'm sorry, 1 or 2. So they're coming toward that end and thinking about sustainability plans. And if there'd be things we could do there that would really boost the value of all of these atlases and collections, that could be, and that'd be a very good thing to do, especially the TMCs have a pause on producing data. Now, while I was sitting here in this meeting, I've also seen other data that is happening just around COVID-19 that we could also think about incorporating.
Yeah, and that's additional data, right, and money right there, yeah. And I think the heart, the kidney, and the lung, all 3 organs, and maybe others as well, are all relevant for COVID.
Absolutely.
Are there other organs besides those 3? See?
I think the data are still emerging. I would say include vasculature with heart, but I think those would be the 3 main.
Yeah, if I can remember what the chart was that they showed the first day, there were a couple other organs, but they didn't they didn't have all the what's the word I'm looking for? The expression wasn't found in all the items they were tracking weren't expressed in all the organs. Some had only 1 and some had, 2, but these ones were the ones that I think had 3. But I mean, actually, in general, though, using the HubMap data for, to look at effects of viruses may be an interesting general problem that, would open up funding from other areas.
Mm-hmm. And we would have very concrete medical problems to solve, Nick, which I totally agree. I think it's great to have a very concrete use case.
Right. So the logic there is viral receptor, in this case, a series of receptors are the markers that you use to probe the pathways, the cell types pathways, et cetera, that are candidates for future work.
Mm-hmm.
And help you develop null hypotheses, which I think was Aviv's summary. Okay. I think if I've got the timing right, we're close here. Everybody happy with the blue box as it stands? Under additional expertise, we need to develop that based on the
Who's the squirrel and who's the elephant?
We can't see ourselves, what we are.
Yeah, if you're typing, you can't see who you are.
Okay, the squirrel is up in recommendations, the elephant's down in expertise.
So I'm the elephant.
I'm recommending.
Okay.
Based on inspection of organs, datasets from CCF Breakout. I suspect we're getting near last call. Any final objections to that?
I'm okay with it.
Okay. Jim, if I might speak out of turn here, I mean, you were saying earlier, well, I'm not a data person. That's not true. You obviously are. You obviously are a data person. And for these things, concrete and defined use cases, as opposed to, oh, we'll make a standard because a standard will make people happier and life will be better for them. I think absolutely critical.
Right. Yeah, I wondered if we wanted to put in the recommendation that, possibility of creating a use case, depending on what other groups are discussing, perhaps, so that we can have something to work on.
Have I articulated that correctly? I guess I'm still the elephant typing under recommendations. And whoever, I'm not sure, Dumbo octopus.
That's a new one.
Questions, probably. Okay. Happy?
Yes.
Well, thanks for your work, Jason.
Thanks. I think you guys did more of the work.
So Nick, are you going to do a soapbox then for all of us, or Jason? Anyone?
I'm willing if no one else prefers to do it. I'll climb the box, but if someone else would prefer, that'd be okay too.
I think it's clearly a good idea, and well, sorry, we're biased, but anyway, that's probably a 30-second soapbox, right? So do it. I think that'd be great. Cool.
So, am I drafted, or would you like to do it?
I'm happy to do it with you. I don't think that means any. I think it means anything. Why don't you make sure that? Please do it, because I think you proposed the target here, the proposal, and you know most about it. I have definitely heard of the CFDE. I've tried to get my head around what it is and what it will achieve a few times, and then kept getting distracted with other things. So.
Okay, well, we'll take a stab at it.
I think you should do it.
Just figure out the mechanism now on the. I guess we just grab the title of this session, the how can we analyze distributed data sets?
Yep. Sorted.
Does anyone else want to be co-listed?
Sure. You're welcome to list me. That's fine.
Okay.
And I have
committed.
Yeah, exactly. So I've written down notes that are confusing me. We can meet back in plenary here in.
I think we are supposed to be there at 2:55, right? So we should go over.
Yeah. So we missed that. Well, okay. We're
well
late.
Done,
okay.
Jason.
All right. Good job. All right. Thank you guys.
You.
See you
Bye.
then.